Hello, welcome to 13 Universal. This is A-level CIE Biology, and we are on planning, analysis, and evaluation, paper 5 of A2 component, all right, A2 syllables, and this is schedule with you. So I will be your tour guide as we take adventure. We want to take our journey into solving questions. So get ready, guys. Bring your writing materials, calculator, your pen. Let's go. So let's go. Now remember, the first thing you have to note is that if you are solving paper 5 or paper 4, any A2 component, you must use a massive reading style. That means as you read, think alongside. And do not forget everything you read, because they serve as clue. They serve as clue. We call them stimulus materials to help you know how to think in order to solve questions. So let's go quickly. Now, the first one says potatoes are underground organs made by potato plant, Solanium tuberosum. Fig 1.1 shows a potato plant. That's it. Wow, that's potato, sweet potato. Is it sweet or Irish? I like this. Looks like Irish potato, okay? But I love sweet potato. So this is it. Is a tuber plant, okay? Now underground organ, okay? You can see that. See it. So, but let's hear what they have to say about it. So, potatoes contain high quantities of starch and a popular food. People love it. After harvesting potato, potato are stored. During storage, the starch content of potatoes gradually, now pay attention, gradually decreases due to the breakdown of starch molecules into glucose. So this is very important. Don't forget it. So it gradually breaks, starch decreases to form glucose. A glucose assay using a Benedict test and the calorimeter can be used to determine the concentration of glucose in potato. So an assay is going to be used to determine that. So let's find out how it works. So look at that's all about long read. So read and remember, long read, guys. The glucose assay uses these steps. These are the steps it uses. The outer skin is removed from potato. The potato is cut into small pieces that are then ground into a pulp using a mortar and a pestle. The potato pulp is filtered to remove most of the solids and obtain potato juice. Excess Benedict solution is added to the potato juice. The mixture is heated in a water bath at 90 degrees Celsius for 5 minutes. During this 5 minutes period, copper 1 ion, so copper 2 ions, copper ions in the Benedict solution react with glucose in the potato juice to form an insoluble precipitate. Okay, that's what is formed. The mixture is filtered to remove the precipitate. The filtrate, which is blue, so remove the precipitate, then you take your filter, which is blue, contains copper ions that did not react with glucose, okay, in the potato juice. Now think a minute. Copper ion that did not react, that means that the concentration of glucose, is it high or low? Why do we have copper ion that did not react with glucose? That means that we have low concentration of glucose. Okay, so we still have excess copper ion. All right, so what do we do with that? Right, see what they say here. A sample of the blue filtered is transferred to a colorimeter tube that's coveted. A colorimeter is used to measure the absorbance of blue filtrate, okay, which is due to the presence of copper ion. All right, a calibration curve is used to determine the glucose concentration of potato juice from the measurement of absorbance. Okay, so measure absorbance by using standard solution, and then you can use it to estimate the concentration of this unknown glucose concentration from the potato juice. Come on, you can read this again and again, but come, let's do something here. Look at it, guys. Let's solve questions. So fig 1.2 shows one type of colorimeter. This is a type of colorimeter. Hold, okay, hold the colorimeter tube, which is coveted. That's it here. That's the digital readout, absorbance, okay. So like I said, every diagram. Every text is a stimulus material. Whatever you are given helps you to understand that. Okay, now let's go to the next question. Is it a clean colorimeter tube containing the sample to be tested is placed in the colorimeter? All right, it's placed here. This is where it is placed, and then light is passed through the sample. So light passes through the sample, okay, and then the absorbance of light by the sample is measured. So as light passes, light of a particular wavelength, all right, remember that what this has here, guys, the secret here, okay, what is here is the blue color, all right, which is due to the concentration, due to the presence of copper 2 ions, right? So the copper 2 ions absorbs a particular wavelength. So wavelength is blue in color. Of course, that wavelength should be maybe yellow. Okay, all right. If you use color rim or color circle, if you so the one opposite blue is yellow. Okay, so that means that this copper ion absorbs yellow. Let's assume. Okay, that's what it should be. And then of course that is the one we should use. Okay, that means you make sure that the light you are passing is yellow. So if you pass white light, normal light, you must use a color filter that allows only yellow, because that yellow is the one absorbed by the copper 2 ions. Okay, use wavelength that is absorbed by the chemical in the solution. Okay, that is where you need to use a color filter. All right, let's go on, guys. We don't have time. Now the first question there, outline how colorimeter should be prepared before carrying out measurements on samples so that correct absorbance readings in the glucose assay are obtained. Pause this video and try to solve it. Okay, I've already given you a clue. All right, this is just about 1 mark or 2, okay, 2 marks kind of. How do we prepare our colorimeters? Now the first thing you have to do is you have to set the wavelength. How do you set the wavelength? You have to set the wavelength. Okay, set the wavelength of light you are using. So you use color filter that allows only the color, the wavelength that is absorbed by the chemical in the solution, and that chemical in the solution is copper 2 ion. There's a particular wavelength that it absorbs, so use a color filter that allows only that wavelength to pass through the solution. Okay, so use it to set the wavelength. But another thing you have to set, guys, is not just wavelength, you have to set absorbance. So you have to set absorbance, you have to calibrate your colorimeter, okay, to 0. All right, remember that absorbance is a measure of the amount of light absorbed. Absorbance is the measure of amount of light absorbed. So the more chemical you have, all right, the more a particular wavelength will be absorbed. So to get 0 absorbance, use distilled water. So use distilled water because distilled water does not have any chemical. So use distilled water to calibrate your colorimeter. All right, yeah. So let's see what the mass scheme says here. So anyone from the following, all right, yeah, use color filter, set wavelength. So color filters, it only allows the wavelength. Remember that light has many wavelengths. Okay, we cannot use wavelength that is not absorbed by the chemical for absorbance. You use the one that is absorbed, okay, to know how much of it that is being absorbed. So color filter is used to allow only the wavelength that is being absorbed to set wavelength of the light. Okay, that's one of the things you must do. Calibrate colorimeter. We'll set colorimeter absorbance to 0. Okay, and how do we do that? Okay, which liquid gives us 0 absorbance? The one that doesn't have chemical, and that's use distilled water to calibrate colorimeter to 0. Okay, guys, let's go. Just 1 mark, but they ask questions a lot from this. A student was given a 2.0% stock solution of glucose from which to prepare a range of standard of glucose solutions of known concentrations. Remember, if you're going to plot a standard curve, you must use solutions with known concentrations. All right, so you can plot a curve between that concentration and what? And absorbance. All right, so that we can then use the curve to estimate or determine the concentration of a known or unknown solution. Okay, now you can see that state the glucose concentration the student could use to produce a calibration curve. Yeah, state it. State the glucose concentration from this stock, produce other concentration. That's what they're asking. And this is embodied. Describe how 20 cm cube of each solution should be prepared by proportional dilution. Proportional dilution is same thing as simple dilution of the 2.0% stock solution of glucose. So this is asking you for dilution. Okay, in our previous videos we've done that, but we'll do it also here. All right, so the first question they said you should state the glucose concentrations the student could use to produce a calibration curve. Now pause the video and try to state it. From 2%, guys, which other concentration can you form? All right, from 2%, which other concentration can I produce? Which other concentration? I have 2% already. Okay, which other concentration? Remember, the principle is that you must find concentrations between 2 and 0, right? Between 2 and 0, yeah, and they must be extra 4 added to 2. Okay, so this is a range. In determining that, you have to know the range. The range is between 2 and 0. Okay, so that's the range. The range is always from the stock concentration to 0, right? You have to find other concentrations between them, and of course you must have a common interval. So interval is the number in between them. What's the interval? The number, the value in between each concentration and the next concentration. That's the interval. You must have a common interval, all right, that is spaced out. Now I can choose now 2. I decide to remove 0.4, so the next one is going to be 1.6. all right, I just want to choose something that will give enough spacing, and it will give me enough value that runs from 2 to 0. So this is 1.2%. okay, and then remove 0.4 from 1.2, 1.6. all right, it's going to be 1.2. okay, yeah, 1.2%. I'm generating those concentration. Remove 0.4 from that, that will give you 0.8. okay, 0.8. So somebody will be asking, how do I know 0.4? okay, now just find something that spaces out. all right, if you choose 0.2, it may not give you enough space. If you choose 0.5, so you try 0.4. all right, and then remove this is going to be 0.4. 0.8 minus 0.4 will give you 0.4. okay, 0.4. yeah, let's see how it goes. So yeah, sorry for the delay. So we have our five concentration, right, including the stock. You must have 4 extra plus the stock. So I've done the first one, this first question that says state this. I've done that. Say the concentration the student could use to produce a calibration curve. Okay, I've done the first part. Then the second part, which is from here, that second part which says describe how 20 cm cube of each solution should be prepared by proportional dilution. Now, of the 2.0% stock solution, if you want to do this, guys, use table. Okay, table is very important. So first of all, draw a table. all right, you must have to draw a table that will help you to organize. Let me draw a table. The table, remember, the table must be enclosed. So we prefer enclosed tables, right, in biology. Okay, I think in all scientific experiments. Okay, so this is enclosed. Now have heading. Now this is the heading. Okay, I should have 3 columns for the heading. Yeah, this is the concentration of the stock. This is the volume of the stock, because we're going to dilute. You need volume of the stock, and that is for water. Okay, I think I should shift it. Yeah, let's move this a bit, and this a bit. This is much. Let's do it again. all right, yeah, just follow through to the end of this video. You're going to learn a lot. all right, yeah, this is paper 5. So this is it. Oh, okay, not well balanced. So this is going to be the first rule, concentration. I choose it, concentration. So concentration, I don't want to write the full this, but I can write it, of stock solution. Of course, different concentration of stock solution, of stock, or concentration of glucose. You can say concentration of glucose. Yeah, let's use glucose, because even the stock came from glucose, right? Of glucose, or reducing sugar, of glucose, right? Yeah, glucose. Do you like glucose, guys? Glucose. Now, of course, concentration is the first one here, but you must add the unit. Okay, that's concentration. Is measured in percentage only on the heading. Okay, you can put this like this, and then this is the volume of the stock. So because you are going to dilute others from the stock, this is proportional dilution. So volume of stock solution, that's your V1 for each of them. Okay, that's volume of the stock here. Sorry for slow writing, right? Okay, that's volume of stock, S-T-O-K, and that is in cm cube. The unit is in cm cube. All right. Yeah, then we have the next column is volume of water. So I can call it V2, but in exam, please write volume of water. I'm just giving a short form. So volume of water, that's V2, the volume of water, and that is in cm cube, volume of water. All right. So let me draw the cells. I have 4 concentrations of this is 4. Okay, 5 concentration of fifth. Right. So this is it. Oh, clean up this. This is the first one. Okay, this is the second one. So I'm drawing the table. This is the third. Okay, because I have the fourth. Okay, maybe the fifth and the sixth. The sixth should be 0, which is our control. All right. So this is 2, right? This is, of course, this is going to be 1.6. That's the next one, right? Okay, this is going to be 1.2. We're doing justice to this question. 1.2, and this is going to be 0.8. So keep minusing 0.4, 0.8, right? And then we have 0.4. So 0.8 minus 0.4, that's 0.4. Okay, and then the last one is 0. This is 0.4. This is 0. Okay, control. All right, now, of course, because this is 20 cm, because each solution will be 20 cm, 20 cm cube. I'm not adding water because it's my stock, right? So from that, remember that has given me the final volume. Final volume, all right, V2 is always V1. V1 is the stock volume of the stock solution you're diluting plus V2. Is that okay? Plus V2. All right, that's, sorry, plus volume of water. Okay, so yeah, that's my final volume, guys. So my final volume here is already 20. Okay, because this is volume of stock, which is V1, plus volume of water. 2 plus 0 is 20. I know that my V2 is already 20. Okay, so my V2 is already 20. Okay, now, but guys, that's 20 cm cube. 20 cm cube. Remember what the question says. We're still on the question. Remember what the question says. The question says we should, that's the second question there. We have not forgotten what it says. Describe how 20 cm cube of each solution should be prepared by proportional dilution. Okay, so we're going to describe it. Of course, we're going to describe it. You're going to take, all right, the table will show us how you did it. Okay, so it's good. Set up the table, all right, or you just pick one of them and then describe how you did it. Okay, so I'm just going to pick one or two. Remember that this is our stock. This is the beaker containing our stock, which is 2%. I'll just write 2 here. So we're going to produce the next one, concentration, which is 1.6 from it. Okay, so from the same, because this is proportional dilution, so we produce all right, every other concentration from the stock concentration. This is 1.2. okay, all right, and then and on. Of course, the next one is going to be what, 0.8 and all of that. Let's not waste much time. Okay, so but I'm just using this opportunity to teach you this. All right, so that means if I'm going to produce this, I'm going to move from here to here. All right, so the V1 for this, okay, that's the volume of the stock, which is 2%. Then I'll add water, right, and make sure I make it up to 20. So this is volume of water, okay, that should be added. Volume of water, H2O, okay, yeah, H2O. So if I want to produce 1.2 from 2% stock, this is proportional, so you use stock to produce every other. But if it's in serial, you don't do that. You use the immediate, all right, preceding solution to make the succeeding. Okay, now add water, just like that. Okay, so you need to calculate V1 for each of them. All right, yeah. So if I'm going to make 1.1 from 2, guys, this is taking time, but it's okay. Remember that you always need to calculate your V1. So from that formula, C1, remember C1, okay, V1, that's dilution formula. Sorry, V1, not V2, okay, equals to, okay, C2, V2, C2, V2. So we're looking for V1. V1 is the volume we want to dilute. Okay, the volume of stock solution we want to dilute. So our V1 here, guys, will be what? It will be C2, all right, V2 all over what? C1. So if I want to do the first one without wasting time, my C2, our C2 there will be 1.6, because that is the final concentration, okay, times V2 is final volume, which is 20, because they said that each solution should prepare 20 cm cube of each solution. All right, that's V2 divided by C1, which is stock, which is 2. Okay, so by the time you multiply out, 1.6 times 20 will give you 32 divided by 2, that's 8 cm cube. Okay, punch your calculator. So that's my V1 here. Is that okay? So I know to produce 1.2, I'm going to take 8. All right, I'm going to take, okay, is it 8? This is going to be 32. No, it's not 8. Yeah, so 1.2 times 20 will give us what? 32. 32 divided by 2 will give us 16. Yeah, that's it, 16. Good. So I will take 16 of stock solution. So that means I'm going to take, sorry, I'm going to take 16 of the stock solution. All right, so that means that this V1 here, yeah, this should be 16, this should be 16, right? So I'm taking 16 of 2%, right? So that means I'm taking 16 of it. How much of water will I add? So if I take 16 of this concentration, right? So I take 16 here, yeah, 16 V1, because I'm moving from here to here. Take 16 of these, then you have to add, how much of water do you add to make it 20? Okay, that's 20 minus 16. I'll add 4 cm cube of water. So that means volume of water I'll add is 4. Now if I'm moving from 2 to 1.2. So that means my final concentration is 1.2. So it's going to be 1.2 times 20 divided by 2. I just remove this and put 1.2. Okay, so 1.2 times 20, all right, is right 12. No, it's 24. 24 divided by 2 is 12. Okay, yeah, because watch it. If I'm moving, guys, please don't misunderstand this. If I'm moving from this to this is my C1, this is my C2. So anywhere you see C2 here, you put 1.2. Everything is constant. V2 is constant, C1 is constant. So that's going to give me it's going to give me 12, right? So I'll put 12 here. Okay, 12 here. And then what do I put here? 12 plus what will give me 28. So you can see the trend. Can you see the trend? The trend is that I remove 4 from 6. Remove 4 from 20, get 16. But remove 4 from 16, get 12. So I don't have to continue to calculate it. So this is going to be 12 minus 4 is going to be 8. Okay, now add 4 to that. That will be 12. Okay, that will be 12. So remove 4. This will be 4. Okay, this will be add 4. That will be 16, right? Of course, to produce this is going to be 0. Okay, so it doesn't have glucose at all. That's 20. Okay, guys, so they say we should describe it. I can write a table for them, describe one of them. That, for example, to make 16%, I'll take 16 cm cube of 2% concentration, add it to a beaker, and then now add 4 cm cube of water. Okay, we have done this, guys. You can replay this video, but let's see the answers. Okay, you may use the table for a concentration. These are concentration for the first question: 2%, 1%, 1.6, 1.2, 0.8, 0.4. Pardon me, I want to clean this up. Okay, yeah, let me clean it up. Let's clean up the table so we can see the masking. All right, yeah, okay, but I hope you learned it. This is proportional dilution. In proportional dilution, all right, the stock solution is what we dilute from. So C1 becomes constant. We keep calculating for V1 because C2 is always V2 is always constant. Okay, all right, yeah, so let's see the answer. Say answer. See now, guys, this is just 3 marks. Everything we have done just 3 marks. It's a 5 stated. Look at it, 5 stated concentrations and units. So 2%, 1.6%, 2% here, 1.2%, 0.8%. So units must added, emboldened, reasonably evenly spaced. Yeah, 2, 1 point. There's even spacing. Method of for dilution: at least 2 concentration other than. So you describe it, right? How you produce. Okay. Sketch on Fig 1.3 a graph of the expected calibration curve that the student would obtain. All right, include the axis. Yeah, sketch it. So remember, what we are doing here is that we're measuring concentration. We're already varying concentration. So concentration is our independent variable. So concentration of glucose, right? Concentration of glucose, okay, fully written, the unit, okay, on x-axis, because it's our independent variable is the variable you are changing. We are changing the concentration. That's why we diluted, and it must be on the x-axis. And then we measure the absorbance. So absorbance is the dependent variable. Dependent variables should be on y-axis. Okay, so this is absorbance. You get? Okay, that's absorbance. Yeah, then the unit there, maybe arbitrary unit. Okay, there's a sketch a graph of expected. Now remember that the more of calcium, copper, ion we have in the solution, the more of a particular wavelength that will be absorbed. So absorbance is directly proportional, okay, to concentration. So it will continue absorbing, all right, until it reaches the peak. After that, okay, it will stay constant because the colorimeter may not measure beyond that, okay. All right, it will see start giving us the same absorbance, but initially that's a positive correlation. All right, okay, let's go to the next question. All right, students decided to investigate the effect of storage time on the glucose concentration of potatoes. Identify the dependent variable in this investigation. Pause this video. What is the dependent variable? Okay, dependent variable is what we're going to measure. Independent is what we are changing. So what are we changing? They said the effect of storage time on glucose concentration. So that means storage time is affecting glucose concentration. The more time we store it, remember that during storage, we were told at the beginning that during storage, starch is broken down to glucose. So that means the more we store it, the more the concentration of glucose. All right. So which one is affecting which one? Independent is one affecting dependent. Which one is being affected? Glucose concentration is affected by storage time. Okay. Which one is going to be measured? We determine this, but this is measured, glucose concentration. So the one that is measured is the dependent variable. Okay. So glucose concentration is dependent variable. Just 1 mark. Let's run. The student was provided with freshly harvested potato, standard laboratory apparatus, and a colorimeter. Describe a method. This is a method. Remember, in paper 5, the first we are still working on question 1. The first question is always on planning, which deals with handling, defining the problem, and then a method. Okay, so method always comes out. It has the highest mark in each question. Describe a method using the glucose assay that the student could use to investigate the effect of storage time on glucose concentration of potatoes. Your method should be set out in a logical order and detailed enough to allow another person to follow it. Okay, detail of how to carry out glucose assay and how to prepare and use colorimeter should not be included. Okay, so this is about 7 marks. Now, anytime they ask you this, okay, they want you to write an essay, a descriptive essay, okay, in a prose format, okay, sequentially. But before you start writing, remember the law of writing in English. You must have an outline, okay. So what should be in your outline is, first of all, you must identify your independent variable IV, your independent variable. Okay, which one is my independent variable? How am I going to vary it? Because what we do with independent variable is that you vary it. You have to specifically, all right, describe how you're going to vary your independent variable. Okay, and then your outline, you already think, okay, this is how I'm going to vary it. For example, the independent variable here is storage time. Okay, so I know that I'm going to have to vary the storage time. So I said, okay, so what's going to happen? I'm going to choose 5 storage times, okay, in days. Okay, I'm going to choose the first storage time I'm going to use to measure is 2 days after harvesting, and then remember, there must be the same interval. 4 days later, I will determine glucose concentration and determine it in 2 days, 4 days, and then 6 days, I'll determine, and then I have 8 days. Okay, these are storage times, and then the last day I'm going to look at is 10 days. So already planning. This is the values for my independent variable. So how am I going to determine? So I make sure that I store the glucose, all right, not the glucose, the potatoes, harvested potatoes, all right, are stored in the same condition, low condition, all right, low temperature, okay, and of course no light there in the dark so that it doesn't start growing, okay, and of course low CO2 concentration. All right, so I store them in the same concentration. Okay, so that's my independent variable. Okay, and then remember, I'm planning to write this. So this is my outline. So I think, okay, my dependent variable dv, what do I do with dv? My dv, all right, I have to measure it. So my dependent variable is glucose concentration. Okay, glucose concentration. So how do I measure glucose concentration? Right? Remember that I already have a standard curve. Okay, because you have a standard curve, all right, that you have plotted of concentration against concentration and absorbance against concentration. Okay, so I'm going to use it to determine that, but I have to also generate absorbance. Okay, how do I measure absorbance? All right, because absorbance will help me to determine the glucose concentration. So use colorimeter to do that. Okay, you can use colorimeter to determine the absorbance. Okay, or you can decide to use glucose Benedict test, then check time, all right, for the first color change. All right, so now, but whatever you are doing must be in line with what I have said, guys. So that's my dependent variable. Then the next thing is what are my standard, okay, variable? What are things I need to keep constant? Okay, this standard variable SV must keep them constant. So you keep constant. What do you need to keep constant? The potato must be of the same species, right? The of course the storage time, storage condition should be the same. The volume and concentration of Benedict solution should be the same. Do you get? So of course, you know, the time for heating, temperature for boiling should be the same. Of course, yeah. So now, of course, you have to talk about validity. How do I make sure that what I'm doing is valid? So you do at least 3 replicates, okay? And then after your 3 replicates, you have to get replicate value. You have to repeat for each of those um, storage time. Repeat and then determine the mean. Did you get? Calculate the mean. And of course, you have to look at hazard. So are there hazard in this experiment? Yes, I'm going to deal with hot water is a hazard. The risk I'm going to get burned. So you write that. How do you prevent it? Precaution. Okay, you use tongs, you wear gloves. Of course, you're using glucose of concentration, maybe irritant. So you wear gloves. Okay, so this makes up your template. You now have to start writing them line by line. All right, and make sure that you use your imagination a lot. Okay, imagine yourself in the lab and you're the one doing this. Okay, so take potato, all right, and then store them in different, you have different storage time, 2 days, 4 days, 6 days, 8 days. Okay, so for each storage time, you know, take the potato must be of the same, of course, the same species, the same age, right? They use the potato to prepare potato juice. Okay, then when you prepare potato juice, you can then carry out glucose assay, add Benedict solution, and then heat it. After that, filter it, but they say you shouldn't go into it. But carry out glucose assay, and then what the solution produce, you know, put it in the calorimeter, and then measure the absorbance. Okay, so you do the same for every one of them. So just look at it. So look at it. Same stated potato used for investigation, same stated storage conditions. These are things in storage condition, low temperature in dark, carbon dioxide concentration, additional storage condition of potato. These are things. Use minimum 5 different storage times, like I've mentioned, 1, 2, 3, 4, 5. Okay, now it continues. Okay, reference to stated storage times of potato. I've stated it. Reference to same volume of potato juice. Yes, use the same volume of potato juice. At each storage time, measure or record absorbance. Yeah, at each storage time, carry out glucose assay. Repeat at least twice, 3 replicates for each storage. So these are the points. And then, all right, the hazard. Benedict reagent is an irritant, toxic. This is precaution. Write it there. Okay, so it's simple to write the method. Just get your out your outline well, okay, like I've explained, and then follow it, writing a descriptive essay using your imagination, right, to do that. All right, 6 marks. That's a lot of marks, guys. Okay, let's run. When potatoes are cooked at high temperatures to make potato chips, glucose and amino acids react together. Pay attention to this. This reaction is known as Maillard reaction and is responsible for orange brown color potato chips. Okay, glucose and amino acid gives me orange brown color potato chips. Have you ever seen potato chips? Now I can relate. Now I can relate why it is orange brown. Very yummy. Now when, and crunchy too, when glucose reacts with amino acid asparagine, acrylamide is made. Okay, this reaction is shown. So that acrylamide, so make sure you relate with the content of your background information. You relate with it so you don't forget it. Okay, don't make it abstract. Relate with it so you can remember it. Asparagine plus glucose at high temperature cooking 120 gives me acrylamide. That's what makes it orange brown. So I'm connecting it. Okay, now that's 1.2. Okay, low concentrations of acrylamide are produced when potatoes are cooked at high temperatures to make potato chips. High concentrations of acrylamide are toxic to humans. They are toxic and can increase the risk of certain cancer development. Very important. So let's go on, guys. Now, background, too many background information, but it's okay. Enjoy the content. Okay, soak it up. Exposing raw potatoes to gamma radiation. This is another information. Exposing raw potato to gamma radiation decreases the acrylamide concentration of potato chips. Remember that acrylamide, high acrylamide concentration can result to cancer development. So these guys said, okay, let's expose it to gamma radiation, maybe to reduce it. Some scientists investigated the effect of gamma radiation on composition of raw potatoes. The effect, okay, on the composition, whether it decreases the composition. All right, 4 bags of one variety of potato were each exposed to a different dose of gamma radiation. So these are the different doses. Okay, 0 joule per kilogram, 50, 100 per, okay, 150. 3 samples of raw potato from each bag were then analyzed. Remember, they are testing your ability to analyze, all right, and evaluate. Analyze chemically to determine the concentrations, okay, of glucose. Now, these are things they're looking out for, okay? Determine the concentration of glucose, protein, and volatile nitrogen compound as percentage of dry mass, okay? So volatile is there. Volatile nitrogen compounds are present in potatoes. The information. Pay attention to information. Due to the breakdown of some proteins and amino acids, such as asparagine. So when asparagine is broken down, it gives volatile nitrogen compound. That's what it means. Statistical tests were carried out on the data to assess whether the gamma radiation dose affected the concentrations of glucose, protein, whether the gamma is affecting the concentration of glucose, protein, and volatile nitrogen compounds in raw potato, which is related to asparagine. Okay, so let's see more information they have. Okay, that's the data, experimental data. Okay, table 1.1, that's it. So you have to study this. Can you see? Look at glucose concentration, protein. This is mean composition of the raw material sample as percentage of dry mass after exposing them to these different doses. Okay, so you can see potato, as the dosage increases, what happens to potato? It increases, almost constant. All right, fluctuates. Okay, look at protein from 10 points. So you have to know this trend, the trend. Study the trend, the pattern. Okay, this increases, decreases, increases. Okay, just a bit. Okay, but look at volatile nitrogen compounds. Can you see? It decreases. That means as this increases, this percentage, the mean composition of raw potato sample, okay, as a percentage of dry mass for volatile nitrogen compound decreases. So this is affecting this. All right, so this is significant. It's significant at probability, critical probability of 0.05. These other ones are insignificant. All right, so this increases that. Okay, so let's look at the question there. It said the scientists suggested that the gamma radiation reduces the acrylamide content of potato chips by affecting asparagine. Yes, asparagine is a volatile nitrogen containing compound. Explain how the information provided, okay, and the data in table support this view. So when they say explain, you have to bring in some biological explanation to that, okay, based on what they have told you and what you have learned from the class. So if you go back to that table, okay, you can see that as asparagine, no, as the dosage of gamma ray increases, what happens is that the asparagine, the percentage of volatile nitrogen compound, like I showed before, okay, decreases, right? It decreases, all right? But why does it decrease? Okay, it decreases because the gamma ray, they say we should explain, the gamma ray is actually causing the breakdown, all right, of asparagine to volatile compound or protein, right, to volatile nitrogen containing compounds. Do you get? Now, but also if you check, is that glucose, right, is fluctuating. So that means glucose is not being broken down. All right, the glucose is not affected and is not contributing to the nitrogen, volatile nitrogen compound. Look at it. All right, as gamma radiation, those, this is those, not those, increase, volatile nitrogen compound, okay, decreases. We saw it there, you can check it. Percentage volatile nitrogen compounds in raw materials are significantly different. Yes, we have seen it from the graph that the percentage is significant. Check the graph, all right, for volatile compounds in raw tomato, the percentage difference, okay, compared to other ones is significantly different. Okay, now of course, because probability is less than 0.05, so it's significantly different. All right, if you check the table, the idea that no change in percentage glucose, we saw it in that table there. Okay, you can go back and look at it. All right, therefore, see, this is not, they didn't say you should describe, that's why they're adding this explanation. If they said you should describe, we state the trend and quote data, that's all. Okay, therefore, decrease in acrylamide cooked potato is not due to glucose, must be due to asparagine. All right, either that decrease in volatile nitrogen compound at increased gamma radiation doses, that is indicate decrease of asparagine, so less acrylamide is made in the Maillard reaction. So we're connecting it to the beginning background information. So that means as this decreases, the acrylamide produced will also decrease. Okay, let's move on to the next question. In a further study, the scientists investigated whether gamma radiation and hot water treatment reduces the acrylamide concentration of potato chips. 4 bags, so now this time we're using gamma radiation and hot water. So to check whether hot water has effects apart from gamma radiation. So 4 bags of one variety of potato were each exposed at different doses of gamma ray. So this was exposed to gamma radiation and also exposed to gamma radiation plus hot water, the same here. All right, that is a dosages of gamma radiation, all right, being exposed. After exposure to gamma radiation, the potatoes from each bag were cut into this 1 cm³ by 1 cm³ by 5 cm blocks. Potato blocks from each bag were given 2 different treatments after exposure. So treatment 1, 100 grams of potato blocks were cooked in 1 dm³ of sunflower oil at 170, all right, degree Celsius for 5 minutes. Treatment 2, 100 grams of potato blocks were equally heated in 500 cm³ of water at 850 degrees Celsius, 85 degrees Celsius, for 5 minutes, removed from the water, and then cooked in 1 dm³ of sunflower oil at 170 degrees Celsius for 5 minutes. So these are different treatments. You want to see the effect. Okay, the scientists measured the acrylamide concentration of the potato chips after this treatment. This procedure was repeated a further 5 times to allow statistical tests to be carried out on the results. So that is it. The results of the investigation are summarized, table 1.1. So you can see, guys, quickly, this gamma radiation dose, as it increases, you can see that mean acrylamide concentration of potato blocks for treatment 1 and treatment 2, okay, they both decrease. But you see that this decreases more, all right, because we use equal. For example, this is zero gamma radiation. Okay, you see what is left, the mean, okay, is much compared to what is left here. Okay, so this decreased a lot. What is left is higher than what is left, all right, after treatment. So this decreased more. If you check 50, what is left after the treatment compared to what is left here is also this is okay, much less left, which is more left. So you can see that as the treatment, as the gamma radiation dose increases, Okay, there's a decrease, but this decreases more. Treatment 2 involves, all right, the gamma radiation and of course hot water. All right, so don't forget this pattern, right? Okay, look at the table again. Using data in table 1.1, calculate the percentage decrease in the mean acrylamide concentration of potato blocks given treatment 2 compared to potato blocks given to treatment 1. All right, you want to check what is percentage decrease, all right, between these two, right? So what you do is percentage decrease, you can pick any one of them, okay? But we can also, you can say, let's assume that you're starting with this, okay? What's percentage decrease from here to here, okay? So that will be 4, 5, get your calculator or try it, 4, 5, 5, 1 minus 1, 7, okay, 6, 8, all over the original. That's percentage decrease, okay? So the decrease all over the original, which is 4, 5, 5, 1, okay? 4, 5, 5, 1 times 100%. So times, this is times 100%, okay? All right, so if you do that, you're going to get 61%. Try. All right, okay, correct, working. So that's the working there. So 2 marks. All right, let's go to the next one. The result in table 1.2 shown as a graph in fig 1.5, error bars have been added to show 95% confidence interval. Yeah, to show it, that's the error bar. That's the error bar. So you can see the error bar. This treatment of gamma alone. See the error bar here, error bar here, error bar. Okay. This is treatment of gamma and hot water. This is the error bar. So what do you observe about error bars here? So the error bar here, anyway, overlaps with this. This overlaps with this. This also overlaps. No, this doesn't overlap. Can you see? The error bar here doesn't overlap with this, and this also does not overlap with this. All right, that is for treatment with gamma radiation. You have to study your result. Okay, can you see the error bars for treatment of gamma radiation? There's overlap, and you know, whenever there's overlap, then the difference is not significant. All right, overlap between these, overlap among all of them, overlap between this and this, overlap between this and this, overlap between this one and this one. Okay, when we say overlap, what does it mean? When we say overlap, okay, trace the upper limit and the lower upper limit of one. If you want to know overlap, trace this upper limit, okay, and trace the limit. You can see that they overlap, part of them. Okay, here this is the overlap, right? So the same thing. So but let's analyze this. You see that gamma radiation dose. Now for this content treatment, this treatment 1 gamma, you can see that it decreases, right? But this one, the decrease here is more, okay? Is far more high decrease compared to this. But overall, as gamma radiation increases, that's general trend, both in treatment 1 and treatment 2, the mean acrylamide concentration decreases, but it decreases more here, okay? All right, so if you are asked to support this, what supports that, all right, that this treatment works? Because this has more decrease compared to this. It has more decrease compared to this, you can see that, okay? All right, now apart from that, you can see that it does not overlap. Now there's significant difference between treatment 2 gamma radiation and hot water. This on the same dosage, look at it, 0 and 0. This is treatment gamma radiation only. This is gamma and hot water. Now the same dosage, 0 and 0, they do not overlap. They don't overlap. In each gamma dosage, there's no overlap. That means there's a significant difference between them. Okay, all right, that means hot water has an effect. Okay, all right, but you know, what doesn't support it is because if you check, all right, for each of these, you see that they overlap. So it doesn't seem the hot water has effect, because for gamma radiation and hot radiation and hot water, every dosage overlap. Okay, so this does not support it. And if you check this one, also there's a decrease also in gamma only. Okay, so this is analysis. The scientists concluded that the combination of gamma radiation and hot water treatment is the most effective way to reduce their acrylamide concentration. Is it the most effective way or not? Is it? Now, you can support it. It's the most effective way because there's much decrease in gamma and hot water compared to gamma only. Can you see the decrease is quite low? This is this. Okay? Now, and of course, when you check, you see that their error bands do not overlap. Okay, so that makes sense. But if you can decide not to support it, because this also causes a decrease, just like this, gamma only causes a decrease. And if you check here, gamma plus hot water, there is overlap across the dosage. Okay, there's a little difference. The difference is not significant, so it doesn't support it. All right, and there's no statistical test. All right, they say explain how the information shown in table 1.1 support or does not support. So this is evaluation skill. They're asking to evaluate the data. So make arguments of for and against. So any three from support. Either that gamma radiation and hot water decreases the acrylamide concentration more than gamma radiation only. Okay. All right. So we have seen that. You can see that either that 90% confidence, that error bars 1 and treatment 1 and 2, they do not overlap. That support it. That means there's a significant difference between two of them. Hot water actually helps to decrease it. Then you now quote data. But if it doesn't support, look at it. Either that hot water treatment alone also decreases it. And again, if you check the error bar, overlap at all gamma radiation doses. Therefore, that is for treatment 2. I've shown you that. Therefore, decrease in acrylamide due to hot water treatment and gamma radiation is not significant different from hot water alone. So that doesn't support it. Okay, you can see that no statistical test carried out. All right, and of course only one validity for the two. Now, should carry out statistical test. All right, let's quickly go to the next question, guys. Sorry for taking time, but let's rush through this. This is quite huge. All right, but let's rush through it. Come on. Rice Oryza sativa is an important food crop rice plant. Okay, rice plants are wind pollinated pollen containing the male gametes. Okay, look at it. Pollen containing the male gamete is transferred by the wind to female reproductive organs of rice plants. Fertilization and grain formation then occur. So weedy rice is a wide form of rice that grows in field of cultivated rice. So weedy rice competes with cultivated rice for resources. That's like a weed. It's a weedy rice. Weedy rice plants are taller than cultivated rice plants and produce a low yield of rice grain. Several genetic modified GM varieties of cultivated rice have been developed. One concern about the use of GM rice is a gene flow from GM rice plants to weedy rice. Okay, there's a gene flow between GM made from the cultivated plant, right, and the weedy. There's a gene flow. Gene flow occurs, when the wind carries pollen grain from GM rice plant to weedy rice plant. Some scientists investigated gene flow from herbicide-resistant GM rice plant to weedy rice plant. The GM rice plant had a gene for herbicide resistance. Weedy rice plants do not have the gene for herbicide resistance. So GM has the gene for herbicide resistance. Weedy doesn't have it. So as you read, you think through the lines, okay? Think through the lines. Okay, let's go on with the bulky context, background information. The scientists wanted to test the hypothesis that gene flow from GM rice to weedy rice decreases as distance between the GM rice crop and weedy rice increases. This is the hypothesis, okay? Gene flow decreases as distance between them increases. The scientists planted GM rice and weedy rice in a field as shown in Fig 2.1, 52.1 also shows that the wind normally blows in a northwest direction. Okay, that's a default wind direction. All right, look at it. This is a black GM plant. This is a woody. This is a space bear. Okay, so this is GM. So you see that the woody rice were planted, okay, in between at intervals. This is 1 meter from GM. The second one is 2 meters from GM. The third one is 5 meters from GM, and then 10 meters. Okay, this is direction of wind in that place. Normal wind blow. Okay, can you see that? So we are varying the distance of this woody rice from the GM. So we are trying to say, prove this, thus increasing it decrease gene flow. Remember that we're expecting that the gene flow, gene for herbicide resistance will move from herbicide resistance will move from GM to woody rice. Okay. Now, after plants had been pollinated and the rice grains had developed, the grains were collected from the woody rice plant only. Grains were collected from all woody rice plants growing 1 meter from the GM rice plant in the directions north, northeast, east, southeast, all of these directions. Okay. Grains were also collected in the same direction from GM rice plant at distance of, right, all of the same distance, approximately 1,000 grains from each collection point were planted and germinated in controlled conditions, okay, in a glass house. Please always pay attention to background information. The plants were grown for 3 weeks and then tested to determine whether they had the gene for herbicide resistance, to know whether they transferred it. And then Fig 2.2 shows young rice plants 3 weeks after germination. I like science, so this is it. So 3 weeks after germination, all right, this rice formed, all right. Let's say identify the 2 independent variables in this investigation. So independent variable is what we vary. So what are the 2 independent variables? What are the 2 things we are changing here? What are we varying? Remember that we want to measure gene flow. Gene flow is dependent. What 2 things that we varied, which are the independent variables? What are the 2 things? Pause video and try it. Okay, you can see the first one is distance from GM. So distance from GM, all right, 2 meters, 1 meter, 2 meters, 10 meters. Okay. The second one is direction of wind flow. Okay, direction of wind. Can you see? And direction of wind. Okay, different directions of wind. Okay, we have north, northeast, south. Okay, and this northwest. Okay, see it there. Yeah, distance from GM rice plant and direction from GM rice. These are what we changed. Okay, all right. Okay, let's move on. Make sure you remember everything. Some of the young weedy rice plants had the gene for herbicide. Yeah, they picked it. Wow, gene flowed. Okay, or gene, there was gene. All right, herbicide resistant. This gene had carried to the parents of these plants in pollen from GM rice plant. Outline a method the scientists could use in the glass house to determine how many of the young, when they say how many, count number of young weedy rice plants had the gene of herbicide resistance. How do you test how many of them had the gene for herbicide resistance? Your method should not require extraction of nucleic acid. Okay, so pause the video. 2 marks. How many? How do you determine that? Simply, okay, just apply herbicide and count the number of rice that survives. Okay, apply herbicide to young plants. Count the number that survives. All right. Okay. Now the scientists calculated the percentage of young weedy rice plants that had the gene for herbicide resistance. The percentage was used as a measure of gene flow. All right, that percentage was used to measure gene flow, the dependent variable. The percentage gene flow recorded at each northwest collection point is shown in Fig 2.3. Fig 2.3 also shows the percentage gene flow recorded to each distance from GM rice plant at all other collection points combined. So all other points combined, okay, against northwest. Guys, look at it. Okay, now always pay attention to your graph. All right, this is the independent variable on x-axis. This distance from GM rice as increases, This is percentage gene flow. Okay, see that. But here we have two: northwest collection point and all other collection points. Okay, so guys, if I should ask you, right, what is the trend here? It asks you to describe this this data. Okay, what should, or you should describe it or you should evaluate it, right? So the first thing you have to do, right, I think there's a question on that. Look at it. It says state two conclusions that can be made for the data shown. What does this data tell us? Okay, what's the meaning of this data? That's what it's asking you. Conclusion is to check whether your data supports the hypothesis. Now watch guys, can you see for both of them, all right, increase in distance from GM rice plant causes a decrease, okay, percentage of gene flow. Can you see each of them decreases both in northwest and other directions? Okay, that's what it's telling me. That's the conclusion. And that conclusion is that, all right, there is more. If you look at that percentage gene flow, right, is more. There's more percentage gene flow, right, in northwest collection combined, okay, compared to all other collection. Can you see across board? All right, it has higher than this. Okay, that's another thing I can derive from this data. Another thing, you can see that the gene flow, all right, does not for both of them, both not wind collection, it does not pass, okay, 10 meters. Okay, for both of them, but this one is about 5 meters, it's 10 meters, right? So these are the conclusions. Okay, look at it. Good. Number one, as distance from GM rice plants increases, these are the conclusions. Gene flow decreases. Two, percentage gene flow distance, yeah, is higher in direction of normal than the pollen cannot disperse further than 10 meters. No gene flow in 10 meters. These are all right, 2 marks. Okay, see, let's quickly tidy up this. Weedy rice plants have a gene that results in increased height. Okay, increased height. Okay, GM rice plants do not have this gene. So GM doesn't have it. Pay attention to that. That means that weedy rice plants have it. This gene allows gene flow from weedy rice plant to GM. Good. It's not only gene flow from GM rice to weedy. It's also move from weedy to GM. So they can actually share genes, all right, through pollen grains to be investigated. In a second study, the scientists investigated gene flow from herbicide-resistant GM rice plant to weedy rice plant and gene flow from weedy rice plant to herbicide-resistant, okay, GM rice plants. The scientists planted GM rice plant and weedy rice plant in areas next to each other in a field. That's a field. After the plants had been pollinated and the rice grains had developed, 1,000 grains were collected from weedy rice plants at each of 8 sampling sites, and of course 1,000 grains were collected from GM also rice plant at each of 8 sampling sites. All right, rice plant and the GM rice plant at each site. So what is it then? That is it. You can see percentage of gene flow from GM rice plant to weedy, all right, to weedy rice, all right, GM to weedy. That's it. Percentage of gene flow from weedy rice plant to GM. That is it. Okay. So what do they want us to do? Complete table 2.1 by calculating the mean percentage of, okay, percentage gene flow from weedy rice plant to from GM, sorry, rice, that's this, okay, to rice plant to weedy, that's this. Okay, so you know how to calculate the mean. Add up all this, divide by 8. Yeah, 1 2 3 4 5 6 7 8. So add them up. So how do we add them up? So you've got to add them up. So get your calculator. 0.04 plus 0.013 plus, come on, 0.025 plus 0.019 plus 0.044 plus, okay, 0.025 don't waste time, plus 0.063, and finally plus 0.057. So if you add them up, that will give you 0.286. Then divide by 8. Okay, so divide by 8, add up everything, divide by 8. So so that add up everything, that will be 0. Okay, 286. Don't forget how to calculate mean, and then divide by the number is 8, right? Remember that that is the mean. Okay, x bar, that's mean. All right, yeah, so that's mean. Mean is what? X bar. Okay, so you divide it. But remember guys, why we're using this, because you know that x bar, okay, for this type of data is summation of, okay, x, right? X is each of them, add them up over the total number, right? So 8 is total number. So now that divided by 8, so the answer is 0.375. So approximately 0.0, yeah, I think 0.0, yeah, 0 point what, 036. Yeah, that's it. Okay, all right, so the same thing with the masking. All right, the same thing. Well, how many marks is that? That's 2 marks. Okay, yeah, okay, 1 mark, just 1 mark. All right, now they said we're not done yet, we'll soon be done. The scientists decided to carry out a test, a t-test, sorry, to compare the percentage gene flow from GM rice plant to weedy rice plant with the percentage gene flow from weedy rice plant to GM, okay, to check whether there's a single difference between them. The scientists stated a null hypothesis. Yes, there is no difference between the percentage gene flow from GM rice plant to weedy rice plant and the percentage gene flow from weedy rice plant to GM rice plant. There's no significant difference. Then the scientists calculated the value of T. This is the calculated value. That's 9.043. That's the T cal. All right, T cal. Then table 2.1 shows the probability of the table for the t-test. This is the table tabulated value. Okay, now explain with reference to table 2.2 what can be concluded from the analysis of data collected by the scientists. Okay, explain. Guys, remember that how we roll this is t-test, the same for t-test or chi-square test. Just remember that t-test, t-cal, that's calculated value, which is 9 point something there. All right, if that t-cal is greater, if it's greater than t-tab, the tabulated value, then reject the null hypothesis. Reject it. That means reject it. All right, our t-tab is, I think, is 9.043. Yeah, so yeah, 9.043. All right, let's find our t-tab. T-tab is that you have to find degree of freedom. Okay, and then degree of freedom is usually you know how to calculate it, n1, that's population 1, okay, minus 1 bracket, and then you add it to the second population, which is n2, all right, minus 1, whatever you get there, right, to add 2 of them together. So n1 is 8 minus 1. So that will be 8 minus 1, which is 7, okay, plus 8 minus 1, 2. This is 8. Each of them is 8. This is 8, okay, and that is 8, right? Okay, so how do I know it's 8? Count. If you go back to the data, okay, go back, you find out there is 8 of them. All right, so that's 7 plus 7. So our degree of freedom is 14. 1, 4. Okay, so now I'll go to the table, go to degree of freedom, and trace it to critical probability of 0.05. This is 14. This is it. Okay, And if you compare it here, can you see this is for 0.01, this is 0.05. So that 0.05 is, you can see that 2.14. So you can see that that's 2.14. So T tab is, sorry, 2.145. Yeah, T tab, right, compared to T cal. So remember, the decision making is that if T cal is greater than T tab, reject the null hypothesis, and that's what happened here. T cal is 9.043, T tab is 2.145. So this is greater than this. So the null hypothesis is rejected. So there is significant difference. The difference is not by chance. Okay? There's significant difference between them. So you see, so the critical probability is this. Critical probability, all right, is also this. That's for 0.01. So value of this calculated value is greater than critical probability. So no hypothesis is rejected. There's significant difference, therefore, between percentage gene flow from GM rice plant to weedy rice plant and percentage gene flow from weedy rice plant to GM. Okay, that is all they wanted to say. 3 marks, that's a lot. Okay, last question there. So the result of this investigation were published in a scientific paper. A student who read the paper concluded, the student must be studious, that the results showed there were no reasons to be concerned about gene flow from GM plant to Y plant. The guy said there's no reason from the result. Well, so suggest one reason why the results of this investigation should not be interpreted in this way. So if this student is your friend and you are just talking, having a discourse on this result, what are you going to tell the student? Say, please don't consider it this way. Don't interpret this way. Okay? What are the reasons? What are you going to tell the student? All right? Why he should not, all right, interpret the result that there's no gene flow. He's making a quick conclusion early, right? So what are you going to tell the student? So for me, I'm going to tell the student, right, that see the result, the investigation was done once, it wasn't repeated. All right, so it cannot conclude because it's just one investigation that was carried out. Okay, and the investigation was done just in one location. It was not done in other location. And of course, the investigation, if you check, you see that the investigation was, if you check from the result there, for what we've done, it only involves 2 genes: gene for herbicide resistance, all right, from GM, and the gene from, of course, the gene for growing tall. Is it? Yeah, from the woody rice. Okay. So, and of course, so these things have to be done separately, and we need to do more statistical tests. So I tell him not to be quick in judgment because investigation only carried once. Okay, investigation only on wind, so we didn't do it for other ones. Investigation only investigated, okay, only investigated once or one gene, only in one geographical area. As a scientist, only experimental trails without herbicide use. Okay, no long-term data. Okay, so you can see the reasons. Gene flow, of course, you can tell the person don't conclude because there was actually gene flow, all right, from the investigation from GM plant to Y plant because they showed herbicide resistance. Okay, so these are the reasons. Y plants with resistant gene difficult to remove. Okay, now this is where we stop. Thank you for staying this long. Sorry for this. Paper 5 is long, okay, in explaining. All right, see you next episode. This is Shedro. Bye.